Thanks for the nice introduction. I would like to thank SBIA for providing this great platform for us to have a dialogue with our stakeholders. As you know, in recent years, cancer immunotherapies such as immunocheckpoint inhibitors and CAR-Ts have shown remarkable success in clinical applications. However, there are still some challenges. For example, some cancer patients do not respond well to immunocheckpoint inhibitors. CAR-T has not been shown to be effective for solid tumors. However, evidence have shown that these therapies, in combination with oncolytic virus, may provide improved clinical outcome. So today, I would like to provide you a high-level overview of CMC considerations for oncolytic viral product development from a CBER perspective. First, I will give you a general introduction of oncolytic virus. Then I will walk you through the key considerations for product manufacturing and testing. Next, I will talk about the design of shedding studies to monitor the shedding of oncolytic virus during clinical studies. This will be followed by take-home messages. Oncolytic viruses infect and specifically cancer cells, but not normal cells. The mechanisms of action for oncolytic virus include direct tumor lysis and stimulation of the anti-tumor immune response by modulating the tumor microenvironment, a process turning cold tumors hot. And interestingly, some studies have shown that this immune activation triggered by oncolytic virus may not only limited to the injected tumor, but may also have a systematic effect on distal tumors. Most oncolytic viruses are replication competent, so my talk today will generally cover the replication competent viruses. Oncolytic viruses can be naturally occurring. For example, we have unmodified real virus and Coxsackie viruses derived from lab-adapted strands. Some oncolytic viruses are genetically engineered recombinants expressing transgenes to enhance the therapeutic effect. For example, PVEX, which is an attenuated HSV1 expressing human GM-CSF. Some oncolytic viruses are genetically modified to reduce the virulence and promote the tumor-specific targeting. For example, we have live attenuated poliovirus with the internal ribosome anthracites from a human rhinoviruses for reduced neuron virulence. This shows some examples of viruses being developed as oncolytic viruses. As you see, there is a wide variety of viruses from different families. We have positive, negative, and double-stranded RNA viruses, and also DNA viruses of various families. With so many candidates in the clinical development, only TVEC, also known as Immunologic, which is an attenuated HSV1 expressing human GM-CSF, has gained FDA and EMA approval in 2015 for the treatment of advanced melanoma. As reported in the literature review from 97 published clinical studies of oncolytic virus that are available in the PubMed, most of these trials are in the early phases, and the majority of studies are for solid tumors, with only less than 10% are for hematological tumors. Most of oncolytic viruses are administered by intratumor injection or IV infusion. Nearly one-third of the studies are combination therapies with chemo, radiation, immunocheckpoint inhibitors, and others. As you know, the product development from preclinical proof of concept study to the final licensure is a long process. Product safety is really the key considerations for phase 1. As the study progresses into later phases, the expectations for product and process characterization increase stepwise for better assurance of product safety and quality. This is accomplished through implementation of GMP, process and assay validation, and establishment of appropriate release criteria. My talk today will focus on how to ensure this product safety for phase 1 study. Establishing product safety for oncolytic viruses can be particularly challenging because they are manufactured using materials of biological origin, and terminal sterilization is not possible. Therefore, the safety should be established through material qualification, well-defined manufacturing process, and adequate testing of the product. I will cover these 3 parts one by one. The components for manufacturing include cells, viruses, and reagents. Cells may be primary cells or cells expanded from a master or working cell bank. Sponsors should establish their virus banks from well-documented and characterized virus seed materials. We encourage sponsors to spend a great effort in establishing well-characterized and well-qualified bank materials, because once established, these materials can be used for a long time in your product development. For establishment of cell banks, please keep good documentation of the origin, derivation history, and cultural conditions, because this information dictates the type of advantageous virus testing required. For example, if FBS is used, the bank should be tested for bovine viruses. In general, cell banks should be qualified to confirm their identity, safety, viability, and stability. As you see, most of the tests listed in this panel are tests for safety from microbial and viral contaminants, which should include sterility, mycoplasma, in vitro and in vivo tests for advantageous viruses, viral particles by transmission electron microscopy, retroviruses, and species-specific viruses depending on the cell species and reagents used. For virus banks, in addition to good documentation, they should be qualified with microbial and viral safety, identity, activity, and stability testing. Additional product-specific tests may be required on a case-by-case basis. For example, drug sensitivity for HSV and transgene expression for those with transgenes may be required. We encourage sponsors to discuss the details of their bank qualification strategy with the agency during the pre-IND meetings. Reagents used for oncolytic virus manufacturing include medium, serum, supplements, auxiliary materials, and reagents used in downstream purification. Please pay attention to the materials of biological origin, such as serum, porcine trypsin, insulin, transferrin, etc., because these are potential sources for advantageous viruses. It is also important that you use the highest quality reagents available with adequate qualification and the documentation of the source, deviation, and the manufacturing process. So the second part of manufacturing is process. The safety and quality of products should be built into the design of the manufacturing process. In process controls such as the pH, temperature, dissolved oxygen level, multiplicity of infection, the flow and mixing rate are key to ensure the consistency of your product manufacturing and the product quality. When there is a change in the manufacturing process, comparability studies may be required to determine the impact of this change on product's identity, quality, potency, and safety. The third part of the manufacturing is testing. Product testing is conducted at multiple stages during product manufacturing, including the in-process testing, drug substance testing, drug product testing, and stability testing. The in-process testing is conducted during manufacturing process to collect information to facilitate process development and validation for later-stage product development. I want to bring it to your attention that the mycoplasma and in vitro advantageous virus testing should be conducted with unprocessed bulk harvested from the crude viral culture before any downstream processing, because the downstream processing, such as cell lysis, filtration, and purification, will reduce the chance of detecting mycoplasma and viral contaminants. The in vitro advantageous virus testing is particularly challenging for oncolytic viruses because the viral product can cause cytopathic effect in testing cells, which interferes with the interpretation of the testing result. Alternative approaches may be used to circumvent this problem. For example, you may use antibodies to neutralize the virus prior to testing, but please do not use human or monkey antisera. And if you need to dilute the test articles, please dilute as little as possible. Another approach is to test the mock-infected production control cells that are cultured in parallel to the production cells. The production control cells in the parallel culture should be treated the same way as the cells used for the virus production, except that they are not infected. And a third approach is to use NGS for detection of advantageous viruses. But currently, we do not allow NGS to be used as the sole method for the testing. It may be only used as a supplementary approach to the standard AVA testing. For lot release, products should be tested for sterility, identity, purity, potency, and appearance. The identity can be tested with nucleic acid-based or protein-based approaches. Purity is assessed by measuring endotoxin, process residues, and product-related impurities such as non-infectious viral particles and genetic variants. For potency, we always recommend sponsors to use matrix-based approach to evaluate different aspects of the biological activities of the product, such as the infectivity, transgene expression, tumor-specific cell killing, and the induction of pro-inflammatory cytokines. We also encourage sponsors to start working on development of biologically relevant and quantitative potency assays early in the product development, which may be useful for establishing comparability and selecting an appropriate potency assay for lot release in later phase. Appearance should be visually checked for the absence of aggregates and precipitates for each drug product vial. Please also establish product-specific reference standard and use it as a control for assay variability. In your IND, please establish a plan to assess the product stability and the long-term storage condition by checking the appearance, potency, sterility, and quality. And also please assess the stability and accelerated storage and forced degradation conditions. Please provide data to demonstrate the short-term in-use and in-device stability of the drug product during administration, which should be tested under conditions that mimic those during the administration process at the clinic. In addition, an established shipping procedures should be in place to ensure product potency, sterility, and quality during shipping from the manufacturing site to clinical sites. Okay, so we are done with the manufacturing and testing of oncolytic virus product. So now I'm going to talk a little bit about the design of shedding studies to evaluate the product shedding during clinical trials. Shedding is defined as the release of oncolytic products from treated subjects through excreta, for example, feces, secreta, for example, urine, saliva, nasopharyngeal fluid, or the skin through pustules, sores, and wounds. Please be aware that shedding is distinct from biodistribution, as biodistribution describes how products spread through different parts of the body, while shedding characterizes how the product is released from the body. Because oncolytic viruses are infectious agents, the purpose of the shedding study is to assess the potential for environmental release and transmission to untreated individuals during clinical studies. And the shedding information is part of the product labeling for licensure application. The design of shedding studies should consider the characteristics of the virus. For example, whether it's replication competent, whether the virus can establish a persistent and latent infection, its immunogenicity, and the pre-existing immunity in patients, and the tropism. And it is also affected by the route of administration, dose, and regimen. The shedding data from preclinical studies may inform the design of clinical shedding studies, but they cannot substitute the shedding studies in human subjects. We also encourage sponsors to start collecting shedding data from phase 1 because the close monitoring of patients during phase 1 is convenient for more extensive shedding sample collection. But please be aware that shedding study may need to continue through phase 2 and phase 3, even if you conducted shedding study in phase 1. So how to design a shedding study? First, you should consider what types of samples should be collected. And this is largely determined by the characteristics of the virus, the route of administration, and the shedding and biodistribution data from your preclinical studies. For example, if this is a respiratory virus administered by intratumoral injection into melanoma, respiratory specimens and swabs of injection sites should be collected in addition to other samples. If this is a human herpes simplex virus-based product, samples of mucosal swabs should probably be taken in addition to others. Next, you should consider the frequency and the duration of sample collection. In general, we recommend frequent sampling immediately following the administration. The sampling should be continued until 3 consecutive data points are below the limit of detection. And sampling to capture the second peak of shedding may be necessary. Last, you should consider the method of the detection. We recommend that you detect the shedding using specific sensitive and quantitative PCR-based method, and then you can use infectivity assays to determine whether those PCR-positive samples are infectious. It is also critical to ensure that the assays are qualified and is free from potential matrix interference from the samples. And the samples should be stored and shipped appropriately according to established procedures. Okay, now I have the following take-home messages for you. Product safety is a key consideration in early phase studies due to the replication competency of the oncolytic virus. Because oncolytic viruses are complex biologics manufactured using materials of biological origin, and terminal sterilization is not possible. Product safety should be established through aseptic processing, material qualification, well-defined manufacturing process, and adequate testing. The collection of shedding data in preclinical and clinical studies with well-designed study plan for sample collection and the use of qualified methods is an important aspect of clinical development for oncolytic viral product. These are FDA guidance documents relevant to my talk today. Thank you for your attention. I would be happy to answer your questions.
Thanks, Dr. Liang. So Bo, in the time of a pandemic, that was a question-evoking talk, to say the least. I'm slightly hesitant to ask you this, but inquiring minds, or at least this one, want to know. There was an adenovirus mutant 20 years ago that selectively replicated in p53 deficient tumor cells and killed them, and not normal cells. And at that time, I thought it was pure genius. And that investigator clearly proved himself genius over the next 2 decades. But that oncolytic virus never came to fruition. I don't know the history, but I assume it failed in clinical trials. So what was the mechanistic problem then? And how have those issues been overcome with today's oncolytic viruses?
Hi, Jeff. Thanks for this question. So, I think the product you're talking about is referring to a live attenuated adenovirus with the deletion of E1B gene, and it is a tumor-specific targeting. But the later study indicate that it's not only the tumor-specific targeting is not restricted to the replication in p53 deficient cancer cells, and in the clinical studies, in phase 1 and phase 2 clinical studies of the product, the results looks like it's very promising in terms of its safety and efficacy. But unfortunately, the phase 3 study has not been completed due to because of the corporate financial reasons rather than scientific reasons. So that phase 3 was stopped, and the company decided to change the focus to a small molecule. And I guess the decision made by this company may have been influenced by several factors at that time, because one of the important thoughts at that time is there's a general skepticism about the validity of the efficacy of the oncolytic virus for tumors treatment at that time, and I think this is also related to some limitations of the adenovirus. For example, for that construct, it's not, although there's a clinical potency in phase 1, phase 2, but the replication and the lysis is not as robust as other candidates at that time. For example, the vaccinia virus replicates more robust, and it seems to be more potent and more efficient in lysing than this adenovirus. And also at that time, this virus is not armed with any therapeutic genes as we have now for many viruses. And there is also concern about its biodistribution through IV infusions because of the hepatic uptake of these viruses. And also there are concerns about the immunogenicity of the virus because of the pre-existing neutralizing antibodies and the complements in the serum limit the IV infusion administration for this product. So unfortunately, with the current development in the field, there's more interest in the oncolytic virus because of the combination, the encouraging result with the combination therapies, especially with immunocheckpoint inhibitors. So there are lots of new designs incorporated into not only the adenovirus but also like other novel new candidates of oncolytic virus are currently under development. For example, we have T-VAC that is approved. It's HSV expressing GM-CSF. So many viruses are going to that route to be armed with the transgenes that provide therapeutic effects or enhance the therapeutic effects. So and also I think it's a trend that there's more and more combination therapies with oncolytic viruses, and also we have a lot of new candidates coming up with novel viruses that has no concerns with the pre-existing immunity in human population, and also they are also to evade the pre-existing immunity. There are also novel strategies to hide the virus from the neutralizing antibodies. For example, we have viruses that are hidden in or packaged or like hide in the carrier cells or lipid nanoparticles. So all kinds of new strategies are coming up in this for this field. So I think it looks very interesting and getting much more attention than the field like for a few years ago.
Thanks, Paul. So just along those lines, I mean, I understand the idea that you inject a virus intratumorally, you have GM-CSF or some other cytokine that's being expressed. You know, with the PD-1, you cause an anti-tumor immune response that's then, you know, you get an abscopal systemic effect based on essentially an immunization like. But do you see some of these new viruses, you know, chemotherapy, the reason chemotherapy works is because it gets to every cell in the body. It causes toxicity to a lot of cells, and then it gets to all the tumor cells. Do you see these virus manipulations that they're able to transduce or infect every tumor cell, or is it the fact that they infect a tumor cell and locally they make more virus and then they infect the tumor cells that are around the tumor cell?
Yeah, that's a great question. I think that's one of the limitations for adenovirus also, because there's some data showing that the adenovirus is not as efficient to spread within the tumors when it injected or disseminated into tumors, solid tumors especially. So so that's why there are more new viruses like NDV and Newcastle disease virus and vascular stomatitis virus and vaccinia virus. Those viruses can form syncytial, and that's very easy to pass around inside the tumors to surrounding cells without having without a novel way to get surrounding cells to be infected in the tumor environment. So I think that, and also some of these viruses are being manipulated to. So, for example, the Newcastle disease virus, they can be manipulated to enhance the diffusion activity. So, that is a strategy to enhance its tumor lysis activity in terms of spreading within the tumor.
Great. Thanks, Paul. A quick regulatory question here, which I don't know the answer to, but when you all say, you know, we want the highest quality raw materials, you know, to control the process, but what does that mean? I mean, does that mean a particular company or particular companies or certificates of analysis, or how do I document that I've got the best reagents?
Yeah, that's a great question. So for early phase trials, we don't mandatory that you have to use like clinical grade or GMP grade, but we highly recommend that you find the best quality of the product available in the market. So, for example, the GMP grade and the clinical grade would be great, because this also saves you effort, and because if you are using like research grade, we are going to ask you a lot of questions about the qualification of the materials and the testing of the materials. So if you use those, if you use the highest quality product, so this may save you some trouble in the IND submission. So we for example, if some product use like a human serum albumin, and because this is like human derived materials, if you use FDA approved the human serum albumin, that we won't ask you much questions about the potential of adventitious virus contaminations. But if you use a research grade HSA, and we will ask you to provide information about all these donors' informations and testings to ensure its safety.
Got it. It's worth the money. It's worth the money to get it FDA certified approved. Okay, unfortunately, that's all the time that we have for questions with Bo. Thanks, Dr.